and start this. So, hey everybody, welcome to today's event. This is BioDojo's second public event, so I'm very, very excited to be hosting you guys here. Very kind of a change of gears from our last one. But before we go into that, what is BioDojo? For some of you guys who may have just recently heard about us, we are effectively a community of young people, like mostly around undergraduate to late high school years. We're a group of young people who have basically built a community to kind of support each other. And I guess the uniting factor amongst everyone in our community is that you're either actively engaged in research in some form that contributes into the life sciences field. So whether anything from clinical research to classic wet lab research or like machine learning for small molecule chemistry, anything under the sun when it comes to research for life sciences. We've kind of unified. We were trying to unify students in these spaces who are undergraduates or like high school students, and provide a kind of like a centralized hub of opportunities and mentorship for the people who are interested in specifically going into industry or startups. So we figure that in life sciences education, for those of you guys who are students or those of you guys who were life sciences students at one point, you guys probably have come across this kind of notion that in life sciences, either you go into medical school or you go into graduate school, and that's kind of the dichotomy, or you just leave life sciences altogether. And one of the big founding principles of BioDojo is we want to expose students into the possibility of the bioeconomy. What are the different opportunities that are that wait for science graduates? What does it really look like to join into biotech? What does running about the company look like? Those are questions that we try to answer here in BioDojo for our members. We also put on public events like this where we kind of engage with the outside world. We want to share cool resources, cool information with you guys. And mRNA vaccines have been a very, very hot topic, especially with a lot of questions that people ask: does this integrate into our genome? Is this some sort of conspiracy? There's a lot of misinformation about mRNA vaccines, and it's new technology, which has gotten a lot of people understandably scared. But we think that with proper communication information, something that Wilson's very, very passionate about, science communication, we can kind of bridge the gap between reality and myth. So that's why we're here today. And Wilson, love to give I think Wilson is probably one of the most prolific science communicators I've seen, at least coming from University of Toronto.
I think that's huge, coming from the fact that Ben Ouyang is in the audience here. But okay,
that's true. You and Ben are definitely iconic. I first came across Wilson's unique promo style for PhD defenses with movie posters. So back in his old lab at University of Toronto, Wilson would make these movie posters involving the defense, the committee, and then also the grad student doing the defense. It was amazing. But yeah, so Wilson's an amazing science communicator and amazing scientist. He's now at the McManus Lab here at University of California, San Francisco, working on RNA. So he did his PhD in nanomedicine, and now here he is in UCSF working on nucleic acids. So enough from us. That's a little spiel about BioDojo. That's why we're here. This is why we're here for today's event. Wilson, please take it away.
Thank you, Michael, for that very kind introduction. Thank you for inviting me. As you said, mRNA vaccines is the topic that you've asked me to speak to today. It's a sensitive topic, so I do hope I do it justice. I'm going to cover basically a brief history of the vaccination field. I'm going to move into sort of the nitty-gritty of how we design mRNA vaccines, and then just like the lecture series named Bench to Bedside, I'm going to touch on some of the clinical and practical aspects of mRNA vaccine translation. So I know, oops, can I advance the slide here? Oh, okay. So I just want to give a little background about me, just so you know where I'm coming from. So I did my undergrad in engineering science at the University of Toronto. I did it, and then I went to do a master's in biomedical engineering at McGill University, mostly looking at nanoparticle-based imaging and some cancer nanomedicine. And then I came back to Toronto to do my PhD in biomedical engineering with Professor Warren Chan, doing drug delivery, nanoparticle liver interactions, and with one of my colleagues who is actually on the call today, Yinan Zhang, worked a little bit on nanovaccines, not really on lipid nanoparticle-based mRNA vaccines, but just small-sized vaccines using gold nanoparticles. And as Michael mentioned, now I am a NSERC postdoctoral fellow at UCSF, Parnassus. And in the McManus Lab, I mostly work on viral and protein engineering, as well as gene editing sensors. So, under the broad umbrella of RNA biology. So as I mentioned, this will be the brief overview of what to expect in this presentation. A brief crash course into molecular biology and immunology. I'm going to talk about at the bench, so how to design the different components of the mRNA vaccine, so both the lipid nanoparticle carrier, as well as the mRNA payload inside. And then to conclude, I'll talk about to the bedside, so some practical considerations, including, of course, the SARS-CoV-2 vaccine case study. It's kind of easy to do a case study for mRNA vaccines because there's only one approved one so far. I'm going to talk about some of the caveats and looking ahead, what is next. So without further ado, and crash course. So this is going to be very broad. So I just want to start broad just so everyone's on the same page. So, how do vaccines work? So basically the concept is you inject a patient with some sort of disease agent that is weakened, inactive, or harmless. So to educate the immune system into producing antibodies or some immune response to recognize the disease. And then finally, once this has been achieved, if the patient is ever infected later by the actual disease, their immune system is already educated and can sort of mount a defense against the disease. So even if they do get sick, they don't get as sick and they can recover quickly. So obviously the key here to vaccines is, of course, what can we use for weakened, inactive, or harmless disease agents, right? So here's a brief history of what people have done with trying to make weakened, inactive, or harmless disease agents for vaccination. So the first vaccine is accredited to Edward Jenner, who used a cowpox virus as a vaccine for a smallpox vaccine. So smallpox was a disease that basically devastated back in the 1700s, 1600s back then. It was a very deadly disease. But it actually had a very closely related cousin known as cowpox, which was less deadly. And what Edward Jenner found was that if you infect someone with cowpox, they can recover most likely. And after that, they will be immune to smallpox because there's a similarity between the two diseases that you basically get one and you are protected from the other. So that is the start of vaccination. And then later in 1881, by Louis Pasteur and Jean-Joseph Henri Toussaint, they figured out that another way to inactivate disease agents, in this case anthrax bacteria, is to use chemical agents to sort of kill them. And so that became, known as the first generation vaccine. So you either use a very related disease strain, or you sort of chemically or artificially inactivate or kill the disease-causing agent, and then inject or expose the agent to the person to be vaccinated. So, of course, after first generation, you move on to second generation vaccines. So these are the so-called protein subunit vaccines. One key one was in 1924, which is when people discovered that the toxic compound by tetanus bacteria could be used as just, as an antitoxin and then injected into patients to create an immune response against the tetanus bacteria. And then in 1986, Maurice Hilleman designed recombinant vaccines. So basically you use genetic technology to make an artificial, in this case, Hep B surface antigen. So it's only one protein. And you give this protein to a patient. And from that, the body is educated to recognize this protein. And because this protein is on the surface of the Hep B virus, the patient can then recognize Hep B virus, the whole virus, when it infects a person, and they can get rid of it. So, and then similarly, for the influenza type B vaccine, they use, instead of a surface protein, they use a polysaccharide. So basically a sugar that's on the outside of this influenza virus as the subunit to recognize for the body. And these are the so-called second generation vaccines. And then finally, we move on to the current third generation vaccines. You might have heard of a viral vector vaccine, the first of which was approved for Ebola back in 2019. And then finally nowadays, the recently not fully approved, but emergency use authorization SARS-CoV-2 mRNA vaccines. And these are the ones that sort of encode for a protein that the genetic material for the blueprint for that protein that you can inject into a person, and the host patient can use those instructions to make the actual proteins for immunization. So what really is this mRNA, right? So I just want to briefly talk about the central dogma of molecular biology. So imagine this is the cell, a typical cell in your body. Your DNA here is the genetic code that encodes for every single protein and other things in your body. And it's double-stranded. And then when you make an mRNA, M here standing for messenger, you basically take a copy of the gene, so a short snippet of this gene, and then from the nucleus, it then gets exported into the cytoplasm, so outside of the nucleus, and then using ribosomes, and you can translate. So every 3 nucleotides on this mRNA corresponds to a single amino acid, and then as you traverse through this messenger RNA transcript, you can make a polypeptide, so different amino acids chained together. And then with some proper folding, you can get a functional protein. So this is the so-called central dogma of molecular biology. So when we're talking about vaccine subunits, the first generation, basically, vaccines gave the whole thing, right? You're giving everything. And so it's not very specific in terms of educating the body what to really recognize for. The second generation vaccines gave usually a protein or some sort of downstream subunit. And now this third generation that we're talking about is we're giving mRNA. So this blueprint here, or in the case of the some of the viral vector vaccines, you're giving the DNA. So moving on to RNA and RNA delivered technology, these are the two sort of history. This is the history of how both of these things had to come together in order for mRNA vaccines to become a reality. And in 1961, mRNA was first described and isolated. So it's only been around 50 years, 50, 60 years that we've known about the existence of mRNAs. And then in 1964, Alec Bangham was the first person to make liposomes. He invented thin layer hydration to make liposomes. In 1990, the first experiment to show that if you injected an mRNA encoding for luciferase into a mouse, it is then able to use those instructions to make the luciferase protein. So that was the first time someone had shown that mRNA encoded for proteins, and this could be delivered therapeutically. And then in 1993, using combining liposomes and this technology of mRNA injection, Frederick Martinian designed the first mRNA liposome vaccine. It didn't work very well, but it was enough to showcase the potential of an RNA vaccine approach. And in 1994, we have Austin Bailey and Peter Cullis, both of which are Canadian, I believe, at least Peter Cullis is. This was the first invention of ionizable lipids. And these two people have been in the news very often recently. Catalin Carrico and Drew Weissman in 2005 showed that if you modify the bases in the RNA, you can actually enhance both their efficacy in terms of protein translation, as well as evasion of the immune system. And this was a key sort of discovery in order to enable mRNAs to be translated clinically. And then finally, in 2010, Sean Semple at Cationic developed a second generation lipid nanoparticles that is still in use today for delivering RNA. In this case, they were designed to use siRNA, but the same technology is basically amendable for mRNA as well. And so combining these two discoveries in the last 10, 15 years is what really enables the new development of mRNA vaccines. And so now that you have an mRNA that you can encapsulate into this sort of lipid carrier, how does it actually work? How do we actually get these mRNAs into the body? Where does the mRNA vaccine go, and what does it do? So typically, these vaccines are administered intramuscularly via needle. So you basically inject them into the muscle here. And these nanoparticles can then get into muscle cells inside the muscle. Some of them will also drain to the lymph node and interact with the lymphatic system. And in the lymph node, you have all these different immune cells that will produce different immune responses. So you have, this is just a zoom in or sort of idealized and simplified version of what actually happens in the lymph node or in basically any antigen presenting cell. So the antigen presenting cell is any type of cell that can present antigens and interact with other immune cells, such as T cells and B cells and all those things. But there are also professional antigen presenting cells inside your lymph nodes. So basically what happens is you have this nanoparticle, it enters into an antigen presenting cell. The mRNA is released from this lipid carrier into the cytoplasm. It doesn't need to go into the nucleus because the mRNA is translated in the cytoplasm. So once this mRNA is released into the cytoplasm, it gets translated by ribosomes inside the host cell to create these proteins. And this protein can then be shuttled to the surface of the antigen presenting cell, either directly or through MHC class receptors to typically first to a T helper cell. And these T helper cells then take this and then activate cytotoxic T cells to generate what's called cellular immunity. So it's educating the cell to recognize this antigen and kill anything that has this antigen. Or it can activate and help B cells to produce antibodies against that antigen. And then if you give a second or third dose, depending on your vaccine design, you can induce memory B cells and memory T cells for long-lived immune memory. So that's sort of a very basic and simplified version of what's happening when we look at immunization with mRNA vaccines. So now I'm going to change gears a little bit and talk more about the nitty-gritty of how we actually design all these different components. Because before I've only given you a sort of like 30,000 feet view of how mRNA vaccines work. Now we're going to go into the details. So I mentioned about Sean Semple, Akin Kinnick making the second generation lipid nanoparticles. So what is a lipid nanoparticle? So as its name suggests, it's basically a really, really small ball of lipid or fat. Specifically, it is 4 different types of fat. And these are the ingredients I've listed here. There has to be an ionizable lipid. So basically, it's a lipid that responds to the pH of the environment. When the pH drops, it becomes positively charged. And so at physiological pH, it has to be neutral. That's very important because cationic lipids are actually super toxic in the body. So you don't want to use a permanently cationic or positively charged lipid to make your lipid nanoparticles. They would be very, very toxic to inject into humans or animals or whatever. You have DSPC, which is just basically your filler lipid. It's a very commonly used lipid. You have a PEG lipid, so it's a lipid where there is a stealth polymer conjugated to the surface. And this is sort of to give both shelf life stability to the nanoparticle, as well as a little bit of protection in the body to prevent protein absorption. But in the case of lipid nanoparticle vaccines, because it's intramuscular, the use of a PEG is actually not super important, as important for if you inject it intravenously, where there's more serum proteins interacting with it. And then finally, you have cholesterol, which is used to basically solidify this lipid nanoparticle and to give it more rigidity. And you can see from this cartoon schematic of a lipid nanoparticle, it's not really a bilayer. So it's really actually solid. So you have all of these lipids that are lined up on the outside, and inside you actually have it's completely filled with other lipids. And then within these little domains, you have negatively charged mRNA, that complex inside. And this is synthetically done by mixing these two at certain ratios under acidic conditions, and then in a microfluidic channel so that they mix together to precipitate into these nanoparticle structures. Here's just a molecular dynamic simulation of how it actually looks like. The red is showing you the nucleic acid. The yellow, green, and purple are the different types of lipids that I mentioned before. And so it really does look like this cartoon. It's like, if I had to describe it, it would be actually like chocolate chip cookies within a muffin. And here are just cryo-EM images of how it looks like, where these gold nanoparticles have been conjugated to the RNA. And so you can see that they are basically not, there's no bilayer here. And all of the mRNA is sort of scattered throughout the inside of this solid lipid nanoparticle. So I mentioned this ionizable cationic lipid. Why was it so important that I needed to talk about it in both, the history as well as, before in one of the four components? So as I mentioned before, it is cationic only in in low pH. And inside your cell, when your cell takes up things, it transports them into lysosomes, which gradually decrease the pH of whatever is inside to sort of try and degrade it. So you can imagine if your lipid nanoparticle is being taken up by cells, it's being transported into these lysosomes. If it releases the mRNA payload inside the lysosome, the payload can't get into the cytosol, and so therefore it will never get translated. And if it never gets translated, it's basically useless. You've basically designed a carrier that will get degraded without having done anything. What this ionizable cationic lipid does is, once the lipid nanoparticle enters into the lysosome, because of the low pH inside the lysosome, it switches charge. It becomes positively charged. And when that happens, you change from this sort of really nice cylindrical shape into this cone shape. And this shape change causes the liposome to basically not explode, but basically dissociate from each other. So instead of forming this nice layer structure, you form a hexagonal phase. And so this releases the mRNA that's inside out of the lysosome and releases the mRNA into the cytoplasm for translation. And so here's just what I just described. So you have your lipid nanoparticle being taken up into cells. It's being transported into an endosome that will eventually become a lysosome by lowering the pH on the inside. There's a phase change that occurs because of the ionizable cationic lipid. And so it forms these weird structures where it therefore allows the negatively charged mRNA to be released into the cytoplasm. And so I just want to take a moment to talk about how do we design these ionizable cationic lipids, right? They don't occur in nature because I mentioned to you before that cationic lipids are very, very toxic. So these actually were synthesized synthetically. They're made synthetically. And there's many, many ways of doing them, but it's really hard to know which one will work best. So whenever you have a problem like this, what chemists or lipid chemists in this case usually do is they just make a whole bunch of different lipids. They test all of their different properties and see which one works kind of the best as a screen. So you can see there's like different chemical schematics. You don't really need to know the details. You can just know that there's different branches. There's different chemical moieties that basically affect the pKa. The pKa here is basically the pH at which it transitions from neutral to becoming positive. So the lower the pKa, the more acidic the lysosome has to be before it triggers the charge change. And you can make different lipid nanoparticles using different mixtures of these cationic lipids. And then you can measure how well or not well they release an mRNA. And this is just what's being shown here. Blue is good and red is being bad. And you can also represent this into a chart like this, looking at both expression of the mRNA as well as response of the immune system. Because you can imagine if you have something that's kind of positive, so if you have a pKa that's too high, it's going to be really easy to become cationic. And as mentioned before, cationic lipids are very toxic. You're going to trigger an immune response. So you want to be at some sort of sweet spot where your pKa is low enough to not be positive all the time, but it can't be too low such that your lysosomes can't reach that low of a pH to release the inner cargo. So there's always sort of this Goldilocks regime that you have to figure out. And behold, most chemists have found that an ideal pKa is somewhere between the range of 6.5 to 6.8. And this is sort of the sweet spot for what you want your cationic ionizable lipids to be at. And this is just to show, what is the if you deliver an antigen, what is the immunoglobulin response based on using just this cationic lipids? And you can see that a few of them actually respond quite well. MC3 here is the first generation ionizable cationic lipid. H, M, P, Q, and N are sort of these newer generation ones that people are testing. And I think, if I'm not mistaken, M is the one that is being used in the actual formulations these days. I have it in a slide coming up, I think. Yes, I do. So here are the lipids that are being used by the two sort of leading mRNA vaccine lipid nanoparticles. So of course, there's Pfizer BioNTech or BioNTech. I don't really know what is the correct pronunciation for this. Pick and choose. And the other, of course, is Moderna. You can see that in this Venn diagram, they both use the same type of DSPC and cholesterol. These are basically not changed. There's no patent on these things anymore. So, they can share and no one is going to be upset at one another. BioNTech and Pfizer have decided to use a pKa 6.09 ionizable cationic lipid, which is, kind of unusual based on what I just told you before, where the ideal is at, 6.5 to like 6.9. But there's actually a reason for it, and I'll mention that in a little bit. And their PEG lipid is ALC0159, also a sort of in-house designed PEG lipid. Whereas Moderna uses SM102, slightly higher pKa at 6.68, closer to what you would expect a cationic lipid to be at. And they actually use a now off-patent PEG, which is PEG 2000 DMG. So why do I keep talking about this sort of patenting thing? Well, it's because the patent issues with lipid nanoparticle technology is complicated, to say the least. So here is just sort of a flow chart of who has stake in all of these different patents. The patent in question here is shown on the right. So this is known as patent 069. So it basically defines what a nucleic acid lipid nanoparticle to be. And it lists that, it has to have a nucleic acid. Makes sense, you're delivering a nucleic acid. It has to contain a cationic lipid that is 50 to 65 mole% of the total lipid present in the particle. So that's the ionizable part. It has to have a non-cationic lipid and a cholesterol. So this is the DSPC with the cholesterol. And then finally, it has to have a conjugated lipid that inhibits aggregation of particles comprising whatever. And so this is the PEG lipid. You can see that the patent covers basically the mole percentages of each component. And it's been very, very difficult for different companies to sort of bypass this, because these ratios are basically set chemically so that the most stable lipid nanoparticles are always within this regime. And so if you wanted to make a lipid nanoparticle vaccine, you basically have to play by their rules and, go with this patent. But yeah, it is very complicated in terms of navigating this patent space. So that sort of concludes my statement on the lipid nanoparticle carrier. I want to now switch gears to talk about the mRNA, so basically the payload. And some of you may know, but I just want to go through it just so everyone understands the different components of the mRNA. So mRNA is the messenger RNA, and it's messenger RNA because it has this very distinctive structure and features that allow it to be called an mRNA. The first is the 5' cap. So it's on the starting side of the mRNA, and this basically tells a ribosome where the start of the mRNA is. Then you have the 5' UTR standing for untranslated region. So this is the region that basically allows the ribosome to know where the start codon is. And the start codon is basically the start of the coding sequence. So when this mRNA is translated, this beginning part is not translated. It's only there to sort of index the mRNA and basically tell the ribosomes how often to make this protein and let the ribosomes bind. And then the ribosomes will start making or translating the sequence at this start codon here, going down this full length. And then there will be a stop codon that tells the ribosome to get off this mRNA. and then stop making, so the protein is then terminated. So basically it's the end of the chain for the protein. And then there's a 3' UTR, again, the 3' untranslated region after the stop codon. So again, this is not made into a protein. And this dictates the functional half-life of the mRNA. It tells basically other regulatory elements in the cell how long to keep this mRNA around, or where to transport this mRNA. This is more important in sort of neuronal cells, because you can imagine a neuron is quite, polarized and quite long. So for protein translation, you might want to transport it to, the apical side or the basal side. And this 3' UTR can be used to sort of allow the body to drag it to, wherever it needs to be. And then finally, there is the poly A tail. So the poly A tail serves two functions. One, again, is to give stability to the structure. So it determines the half-life of this mRNA and how long it can be remain intact inside the cell. The other function the poly A tail actually has is, depending on the ribosome structure and the mRNA sequence and the shape, it actually loops back to the front here. So you can imagine this forming a U shape with the ribosome to allow the ribosomes to sort of do this sort of U-turn thing. So it sort of regulates the speed at which a ribosome can zoom through this coding region. And so if you design this correctly with all of these different components, you can actually control how long this protein, how much protein can result from one mRNA molecule. And from there, you can basically increase the potency of the mRNA and reduce the dosing frequency, which is more of the what you want for mRNA vaccine, because you can imagine you can only make so much mRNA, and you can only load so much mRNA into a carrier. You want the most bang for your buck, basically. You want something where the mRNA can make as much of the protein of interest as possible. So here I'm going to talk about sort of globally, just like what results when you translate or when you try to manufacture these mRNA vaccines. So in the ideal world, you would just have pure mRNA product that you've designed, pristine mRNA, encoded in this perfect lipid nanoparticle, and you deliver this into the cell. But obviously, we live in a world where we're trying to make kilograms of this stuff, and enzymes are very good nowadays. We can design even better ones, but they're not perfect. And so what usually happens is you'll get some sort of contaminants. And the contaminants come from this double-stranded RNA or single-stranded RNA that is not what's encoded for, or it doesn't have the perfect sort of sequence that you want. And all of these things can trigger what's known as the intrinsic pathway of the cell, of the immune system. So instead of thinking about the immune system of different cell types, so B cells, T cells, macrophages, whatever, here I'm talking about something that's much more primal, something that's more archaic. It's the immune system within the cell. So when most cells, when they recognize that they've been transfected with RNA, it's usually some sort of virus, right? Because naturally, a virus tries to infect cells, single cells, with RNA. And so this is sort of the single cell level immune response, if you will, to being transfected. Cells in general don't like to be translated. And so you set off this cascade of different proteins reacting, and then you get these like pro-inflammatory cytokines being produced, this interferon response. And so if you design a manufacturing process or some sort of mRNA that triggers all of these things, you're not going to have a good time. You're going to have a lot of side effects from this design. And so you want to avoid that as much as possible. So remember Carrico and Drew Weissman? So their major discovery of how to reduce this sort of undesirable, intrinsic response in cells is base modification. So in RNA, you don't have thymine, you have uridine as a replacement. The cells are actually really good at recognizing foreign uridine. And this is sort of the what they look for in order to recognize, exogenous RNA that they don't want there. And so what Carrico and Drew Weissman did was they methylated it. So they made it into an N1-methyl pseudouridine. So this is still able to be recognized as a uridine in the genetic code, but it has a methyl group on it, and therefore it is able to bypass the uridine recognition of the intrinsic system of the cell. And so what they found was, let me show the data now. So this is quite busy, so I'm just going to go panel by panel. So here they've just made a mRNA that encodes for human erythropoietin, which is a blood protein. U here is uridine. 1M5 is the pseudouridine that I mentioned before. Process A and process B are just two different manufacturing processes. They don't describe in the paper that I'm referencing here what that specific process is, but basically process B is an extra filtration step to clean up your byproducts. And then RNase 3 is an enzyme that removes double-stranded RNA, which is another trigger for the intrinsic system. And so basically the takeaway message here is when you do all of these things in together, so you replace the uridine with a pseudouridine, you do an extra cleanup step with the RNase 3 as well as this filtration step, you can actually remove basically have no interferon beta production in the cell. So you're basically not triggering any sort of intrinsic interferon response. And at the same time, you're still able to make a whole lot of the protein that your mRNA is encoding for. So that's sort of the global view. If we look at the 5' cap and 5' UTR, this is again where the start region is. This is how the ribosome recognizes the mRNA. It recruits both the small unit and the large unit, and this together needs to bind at the start codon. And so depending on how you design this process can be either very efficient or not efficient. And this is an electromicrograph of how it actually looks like. So this is the mRNA chain here. These are all the ribosomes. And if you design a 5' UTR correctly, you can actually load a whole lot of ribosomes onto the same mRNA and therefore get a whole lot of protein production in parallel. But again, this also depends on your sequence. So here in this paper, if you want to read it, there is some machine learning involved. But basically what they've done is they've made a whole bunch of different mRNAs with different 5' UTRs, just like random sequences of 5' UTRs. And through looking at how many holosomes, so basically how many ribosomes can be loaded on, they can figure out what is computationally the most efficient 5' UTR sequence to get the most protein production, or in this case, how many parallel production of protein you can get. So that's one way of optimizing. The other way of optimizing is the actual open reading frame, so the actual protein that you're trying to make. If you use if you have fast translation, this is when you have a codon optimized. So what that means is the human body has different ratios of different tRNAs that encode for different amino acids. And they're in different ratios. So basically what that means is the body prefers certain sequences over other sequences when making protein. If you make something, or if you design a sequence that matches that preference, then you'll get fast translation. But if you design something that has doesn't match the preference, then you'll get slow translation. But it's not just all about the codon optimization, because just because the sequence is what the body prefers, it doesn't mean nature won't fold it into a weird shape. You can imagine that if, depending on what the sequence is, if you design something that has a lot of strong secondary structure, then it's physically going to stop the ribosome from proceeding. It has to unbind this transient interaction in order to go over to the next sort of loop. And so it's a combination of both optimizing for codons and optimizing for mRNA secondary structures. And this is actually how mRNA looks like, or schematically looks like when you make it. It's single-stranded, so it has actually really strong secondary structure features depending on the sequence. Here is the human preference codons. So these are the codons in blue are the ones that the body prefers. Red is the ones that the body does not prefer. And so you can, for the same sort of protein sequence, you can design different mRNAs that have low structure, medium structure, high preference for optimized codons, low, medium, all that stuff. And again, if you do it computationally, you can actually figure out what is the Goldilocks sort of region where you get, good translation, fast enough translation that you get as much protein as you can, but also you don't get these traffic jams where maybe a lot of protein, a lot of ribosomes are loaded and then they're stuck at this secondary structure, and that becomes a limiting factor. So here's just some data showing you that if you optimize for high structure or optimal codon usage, you get a certain expression of green fluorescence. And so this can be done computationally for each protein that you try to make. Looking at the 3' UTR now, I mentioned before that this is important for determining the half-life as well as the subcellular localization. And that's because different proteins will interact with this region to basically transport it along a microtubule or actin, or microRNAs can bind here to stop translation or actually degrade this entire construct. So here is a sort of directed evolution approach to figure out what is the best 3' UTR to use if you want high translation. So what they've done here is in a cell, they've added a drug, the ACTD drug, which stops the cell from making any more new mRNA. And then they just wait. And because mRNA half-lives mean that most mRNAs will have a short half-life, if you wait long enough, only the mRNAs that have favorable 3' UTRs will be left. And so if you wait long enough, you can just collect those long-lived mRNAs, you can make them into a RNA library, and then you can just keep injecting them back into the next cell and just cycling through to see which ones survive even longer. And in the end, you find a certain number of hits. So these are the most frequent 3' UTR motifs that survive the longest. And then once you have hits, what you can do is you can, actually I don't know why these arrows are here yet, but basically you can validate those hits. So you just basically tack on this 3' UTR to a protein of your choice and measure how much of that protein you can make. And if you fancy, you can actually combine these. So if one 3' UTR gives you a certain half-life, what happens if I combine two of these different 3' UTRs? And in this case, you can actually get even more half-life, even longer half-lives. So this is one way of figuring out what 3' UTR you want to use for making your protein of interest. And then finally, I'm going to talk about the poly-A tail. So I mentioned before the poly-A tail determines half-life. It also helps with stabilizing the ribosomal structure to produce the protein. But one of the problems is if you encode for this poly-A tail in your plasmids, there is a high chance of recombination. And what recombination is basically the body or your cells don't like to make these repeated structures. It doesn't like that. And so it basically randomly cuts out things or recombines it with other things. And so you get a very non-consistent number of A's at the end of your mRNA. And from a regulatory standpoint, this is a no-no, right? The FDA is not going to like if you say something like, well, my mRNA has a poly-A tail between 0 to 100, whatever the cell feels like making. And so this has been a problem in the field. And so what these people, this paper came up with was to add a linker in between long poly-A tails. So this is the poly-A 120, so basically 120 A's in the sequence. This is 3x40, so basically you have 40 A's separated by a 6 non-A linker, another 40 A's, and then another 6 non-A linker, and then another 40 A's. So in total, you still have 120 A's. And this is the 2x60, so 60 A's, a 6 non-A linker, and then another 60 A's. So in this experiment, they show that if you break up the length of the poly-A, you get a lower chance of recombination happening. which is what you want. But does that affect protein translation? In this case, in this particular experiment, they show that it actually doesn't. And in fact, it sometimes might actually improve your translation. And you can see here, modification 1 here just is talking about the pseudouridine that we talked about earlier. But if you have a 2x60 poly A separated by a 6 nucleotide sequence, you actually get a lot more protein expression. And finally, I know I'm kind of running out of time here, so I'm going to try and rush through this. I'm going to talk about to the bedside, so practical considerations of how we can translate these mRNA vaccines. And I'm going to start with a SARS-CoV-2 vaccine, case study. And I think I think most of you will have heard the news. This is one of the fastest vaccines to be developed in modern history, ever actually. And that really speaks to the power of the mRNA vaccine, because a lot of this work that I showed before had already optimized for a high expression of a protein. You basically had to only figure out what protein to use for this SARS-CoV-2 vaccine development. And then from there, it was just basically a manufacturing problem and not a design problem. And here is the SARS-CoV-2 genome. It is about 30 kilobase pairs. This is way too long to be encoded for in an mRNA, at least synthetically. The virus obviously is able to do it, but from a manufacturing standpoint, you can't really make this big of a genome as a full protein. So you could not encode for this entire thing in your mRNA vaccine. That would just be insane. And so you had to basically pick one of these proteins. And the obvious candidate is, of course, what's on the outside of the SARS-CoV-2, which is, of course, the spike protein shown here, right? The spike protein is about 1,273 amino acids. So it's within, a reasonable size for an mRNA, a single mRNA to encode for. Going back, there's actually different domains in this protein as well, right? There's the RBD, which is the binding domain of the spike protein. And so, this is just an example from Pfizer, BioNTech Pfizer. They at first tried to make a solubilized small protein. So the mRNA only encodes for the receptor binding domain. And they compared it to a full length spike. So basically the entire 1,273 amino acids. And they actually found that, this still works, but there were some serious side effects in terms, not serious, but like this was, there was more undesirable side effects. And so they decided to go with the full spike length. There's not enough understanding right now to say why that is. But one of the reasons or potential reasons is because this is a solubilized protein. So basically it's being excreted out in large quantities into the body. And so it could be causing some sort of systemic response. Whereas here you have the spike that's tethered to the actual cell membrane. And so it's a little bit more local and more controlled in terms of the presentation of this protein. Okay, so we have the candidate now. So we want to make a full spike protein, but you have to look at the biology. The spike protein actually has a pre-fusion form and a post-fusion form, and in order to make a vaccine that can educate the immune system on how to recognize actual SARS-CoV-2, you want to be able to make the pre-fusion spike shape, right? So that it's the native shape that's on a free-floating sort of virus particle, so that the body can actually recognize the SARS-CoV-2 before it binds to the cell. Because once it binds to the cell, it can potentially enter into the cell and cause disease. So you want to be able to educate the body to recognize the non-fusion form, or the pre-fusion form, sorry. And here's just a schematic of how those two different shapes actually look like. On the left here is the pre-fusion spike, and the post-fusion spike you can see is basically much thinner and longer. And so how do you stabilize the structure? And luckily, we learned from MERS, which is another coronavirus back in, I believe, 2009, Middle Eastern Respiratory Syndrome virus, which is closely related relative to SARS-CoV-2, is that if you make this 2p mutation, so you replace these two amino acids with proline, you can actually lock the spike protein into the pre-fusion form. There have now been more sort of work on this. There are different mutations that you can do, but the current generation of SARS-CoV-2 vaccines use this 2p mutation. And this is just some different cryo-electron microscopy images just to show you that indeed the shape is being preserved when you have the 2p mutation. And, you know, the rest is history, you know, using this sort of approach with the 2p mutation and encoding for the spike protein. Pfizer BioNTech did this clinical trial, and it's just night and day when you compare to the treatment group to the placebo group, right? It's 95% efficacious. So, you know, a success story all around. And now we move on to sort of like, how do you, so now that you have, you know, something that works really well, how do you mass manufacture this? And so the process right now, because of how fast we needed to make this, like as many vaccines as we could, is not optimized. It's optimized enough to make, the doses that we've made, but it's not, super optimized. So there's room for improvement. But basically what you start off with is this plasmid DNA that has your spike protein encoded in it with all of, the 5' UTRs, 3' UTRs that you've designed. You transform this into bacteria. So you let these bacteria take up this plasmid and you let them basically make many, many, many copies of this plasmid DNA. And you basically isolate it back from this bacteria. Then what you do is you subject it to a restriction enzyme digest. So you linearize this circular plasmid DNA into a linear DNA. And this is all being done only in one factory right now, at least for Pfizer BioNTech. And this is being done in, I believe, Minnesota. And then afterwards, you need to actually move this linear DNA to another factory, in this case, Andover in Massachusetts, for in vitro transcription. So basically, you take a virus polymerase that recognizes the a certain start site on this linear DNA, and then from there, it transcribes it into single-stranded RNA. And remember, mRNA has to have a cap, and so you need to do the enzymatic capping reaction to put a cap on. So now it's mRNA. And so this green section is being done at Andover in Massachusetts. And then once you have this mRNA, you transport it to Kalamazoo, Michigan, where you take all these 4 lipids that we mentioned before, you do the microfluidic mixing, bada bing, bada boom, you get lipid nanoparticle encapsulated mRNA coming out of Michigan, a couple million doses every day. Now, how do you transport this now to, where it needs to be, administered? So mRNA and RNA in general, as you might surmise, is super unstable in the sense that it's single-stranded, it has, weird secondary structures, and depending on how you design that secondary structure, if it doesn't have those like loops, it's very susceptible to degradation and different sort of chemical degradation and photodegradation and all those things. And so it needs to be kept in as cold as possible. There hasn't been sort of a systematic study looking at how long the shelf life is of these particular mRNA vaccines. But this is sort of like what the FDA requires right now for storage. And I was only able to find one paper where they kind of looked at how long you can leave an mRNA vaccine out in the open, at 37 degrees, 25 degrees, or 4 degrees for a number of days. And you can see in this case for an mRNA, not vaccine in this case, but a lipid nanoparticle encoding for a luciferase mRNA, you can see that the luciferase production is stable over 7 days if you leave it at 4 degrees, and it gradually decreases over time at 37 degrees. So, it might not be as stable as we want it to be, but it's still, within the realm of possibility of transporting them to different locations around the country, or the world, sorry, in this case. Now, the other thing I wanted to talk about is the PEG dilemma. So you might have remembered back in December, January, when the mRNA vaccines were first coming out, there is this huge debate about PEG and PEG allergies and hyper reactions against the PEG. So PEG is polyethylene glycol. It's only about 0.5 to 2% of the actual lipid nanoparticle. And personally, I don't understand why people are so worried about this, but nevertheless, from a clinical standpoint, it is a concern because there are certain people who have negative responses to or anaphylactic or pseudo-anaphylactic responses to this pegylation. And so it is a problem for a small minority of individuals. And recently a paper came out, a medical case study, looked and proved that one individual who actually had a severe anaphylactic reaction was because of the PEG itself on the Pfizer-BioNTech vaccine. And how big of a problem could this possibly be? Well, an estimate from this study back in 2016 has shown that the potential population that has enough sort of anti-PEG antibodies circulating in their blood normally is about 7%. But you have to remember, this mRNA vaccine is injected intramuscularly, not intravenously. So it's not clear whether or not the intravenous IgG, IgM in the plasma could play a role in the, or as big of a role in reacting to intramuscular injected vaccines. But for sure, PEG is a problem moving forward because even outside of mRNA vaccines, PEG-related protein therapy regularly results in anaphylaxis as a potential side effect. And so what are some possible solutions that we can do? Well, we can actually do a few things. I just list two of them here. On the left here, I'm showing you if you design a PEG lipid that sheds off at different speeds, it's been shown that this can actually modulate the amount of anti-PEG IgM production. And so you can imagine if moving forward, we want to have multiple types of mRNA vaccines using different, the same lipid nanoparticle outer shell, but a different mRNA inside. We want to decrease the amount of IgG and IgM that's created against the carrier itself. And so a fast shedding PEG lipid might need to be designed. The other option is to just remove PEG altogether. PEG is not the only stealth polymer that we can use. In fact, this group in, I believe, Israel made a polysarcosine stealth polymer lipid nanoparticle and show that it has equal sort of delivery potential and functionality as their PEG-related counterparts. And so that's another route that people can go down. And finally, looking ahead, what's next for mRNA vaccines? SARS-CoV-2 was sort of the perfect storm for, getting all of this into the spotlight, but, we've actually had mRNA vaccine trials for the longest time against all of these different sort of viral targets. You can also design them to go against cancer antigens or new neoantigens for personalized medicine and cancer therapy. You can also use them to encode for different immune modulators for tumor microenvironment or immune diseases. In this case, it's not really a vaccine, but it's more of making these cytokines to modulate immune and microenvironment. You can also use them to generate, growth factors for regenerative medicine, and as well as, mitochondrial diseases as well. If you manipulate how that mRNA protein, both the mRNA and the protein can translocate inside the cell. And so the possibilities are quite endless. And so I'd like to conclude with these three take-home messages. Number one, mRNA vaccines are safe and effective. They have been a long time coming. It's taken 40, 50 years of research, bit by bit, to make them possible. We don't understand everything yet, but they are for sure safe and effective in the populations that we've used them in and tested for. Number two, mRNA vaccines have great potential outside of SARS-CoV-2. It's not going to be a one-trick pony. Hopefully there will be more coming along the way after, the manufacturing facilities can take a break or be expanded to, make other things other than SARS-CoV-2 vaccines. And number three, finally, as I mentioned before, there's still a lot of room for improvement to make these mRNA vaccines even better. And, this is just the beginning. We're going to see a lot of second generation, third generation mRNA vaccines in the near future for flu, for, HIV, for all of these difficult diseases that right now we don't really have a good vaccine for. And so I'd like to acknowledge all of these wonderful people on Twitter who took the time to speak to me or educate me on what I presented on today. And I encourage you to go and follow these people or at least check out what they have to say. And I think that's my last slide. Yes. So yeah, thank you for letting me speak to you. And I apologize for going over time.